through this picture regarding structural aspect of endoplasmic reticulum. So here when you take this, assume this as a nucleus part, okay, the central one on this slide. If we assume that this as a nucleus part, from the, yeah, good evening Bhumika, from the outer membrane of the nucleus, which we call as nuclear membrane, you'll have a network of structure, this whole thing. This structure is endoplasmic reticulum. So when you say the position of endoplasmic reticulum would be there from nuclear membrane towards the plasma membrane. Suppose assume that this is the whole cell. Within the cell you are having the organelles. So a cell is surrounded by cell membrane. So from the cell membrane towards the nuclear membrane, you'll have a network of structure called endoplasmic reticulum. Okay. Along with the endoplasmic reticulum, you'll have several other kind of network systems called as Golgi apparatus, which we'll see in this section itself, I hope. Okay. Now, when you see the endoplasmic reticulum, this can be differentiated based on the presence or absence of this bright color vesicles, which are shown in this diagram, called ribosomes. Okay. Now, when you take the presence of ribosome on an endoplasmic reticulum, it would be called rough endoplasmic reticulum. If such kind of ribosomes are absent on the endoplasmic reticulum, like this, it will be called smooth endoplasmic reticulum. So, based on the structural aspect and texture, rough and smooth, which differentiates presence and absence of ribosomes. So, if ribosomes are present, it's rough endoplasmic reticulum. If ribosomes are absent, it's smooth endoplasmic reticulum. So, basically, we are having two types of endoplasmic reticulum, rough and smooth. We come to the terminology of endoplasmic. It's nothing but within the cytoplasm, you'll have an organelle, which is network. Reticulum stands for a little network of thing. Okay, so that extends from the nuclear envelope, which is connected throughout the cytoplasm. And endoplasmic reticulum can be divided into rough endoplasmic reticulum and smooth endoplasmic reticulum. Just terminology, endoplasmic reticulum means a network of cell organelle within the cytoplasm. Okay, that's about the terminology of endoplasmic reticulum. Then this word carries much importance, cisternae. As of we discussed about the structural aspect of endoplasmic reticulum. So in that cisternae word I would have discussed. Now when you take the cisternae, in the later cell organelle concepts, we'll find different terminology related to cisternae. Even the cisternae are present in Golgi complex also. So what are these? These are nothing but a spherical or tubular membrane, okay, which will be there in the cytoplasm and forms a channel of endoplasmic reticulum. See here, if you observe, these are the cisternae. The shaded one I'm showing, those are the cisternae. Okay. Then this is all about rough and smooth endoplasmic reticulum. Presence of ribosomes, absence of ribosomes. That's all. Then coming to the rough endoplasmic reticulum functional aspects, I think we are there here somewhere, synthesis of precursors part, the last session I think.
Yes, ma'am.
Yeah. So this is a common point because ribosomes are present, we are saying, where they will be held by the endoplasmic reticulum. So the space is called ribophorins. Ribophorins are the receptor molecules which will hold ribosomes in rough endoplasmic reticulum. So the place where the ribosomes are attached to rough endoplasmic reticulum, that part is called ribophorin. When you take the point of ribophorin, ribopphorins are the receptor proteins which will hold the ribosomes. Now, whenever we talk about ribosomes, anywhere inside the cell, it's always related to protein synthesis. Proteins will be produced by ribosomes. So, one of the function of rough endoplasmic reticulum, we can say, synthesis of proteins. Okay. Then other than that, the rough endoplasmic reticulum helps in linking of sugar molecules to the produced proteins. Okay, when you take why this linking is going to happen in rough endoplasmic reticulum, that can be explained when we talk about the Golgi complex functional aspect. This point can be related to Golgi complex functional aspect. There I'll clearly explain how the sugars will be linked to the proteins produced in rough endoplasmic reticulum. So for here, about the function of endoplasmic reticulum, especially rough endoplasmic reticulum, we can say they are involved in synthesis of proteins. First point, they are involved in linking of sugars or adding of sugar to the produced protein. So they are called glycoproteins. Here glyco stands for the sugar molecule and protein to the which these sugar molecules are added. So this happens in rough endoplasmic reticulum but gets completed in Golgi complex. So when I discuss the Golgi complex in functional aspect, then I'll explain clearly how the sugars are linked, how the linkage happens. Okay. Then coming to the next function, synthesis of precursors. This I explained by taking example of enzyme reaction. I hope I remember I used the word enzyme precursor when any enzyme, if it is going to act on a substrate to produce a product, before that, that enzyme will be in inactive form. Later, it will change into active form. That inactive form of enzyme we call as precursor. Precursor is nothing but a starting material. Suppose if you take in a crude word, precursor is nothing but a starting material. The enzyme, before getting involved in catalyzing any reaction, that will be called enzyme precursor. Such enzyme precursors will be produced by rough endoplasmic reticulum. Now, if you know, most of the enzymes are proteins in nature. So, as of rough endoplasmic reticulum is involved with ribosomes, so the precursors enzymes production also happens in rough endoplasmic reticulum. Okay. Then about smooth endoplasmic reticulum functions. This we need an elaborate study. Here, when you are saying there won't be any ribosomes, the smooth endoplasmic reticulum doesn't show protein synthesis function. This is the major difference between rough endoplasmic reticulum and smooth endoplasmic reticulum. When you take the differences, the first difference comes as in rough endoplasmic reticulum, we'll have ribosomes. In smooth endoplasmic reticulum, ribosomes will be absent. The second difference between rough and smooth endoplasmic reticulum is rough endoplasmic reticulum is involved in synthesis of proteins, whereas smooth endoplasmic reticulum is involved in synthesis of lipids. So lipid synthesis is the major function of smooth endoplasmic reticulum. And it won't show the protein synthesis because ribosomes are absent. And other than that, smooth endoplasmic reticulum, SER we write in shortcut. When you take smooth endoplasmic reticulum, it is involved in several metabolic processes. What kind of metabolic processes? See here, one thing we said, synthesis of lipids. Other than that, it is also involved in synthesis of steroids and sex hormones also. So sex hormones like progesterone, progesterone, testosterone. These are the adolescent hormones. You're having a topic, I think, in 9th standard regarding. I think in 8th standard CBSE you are having a reaching adolescent stage. There you'd have read about the reproductive system, reproductive organs. There you'll find these hormones. So these are this progesterone is the hormone involved in female reproductive system. The testosterone is the hormone involved in male reproductive system. So such important metabolic processes involved hormones are produced by smooth endoplasmic reticulum. Okay, so here it synthesizes lipids, steroids, and sex hormones also. Along with that, it helps in breakdown of glycogen into glucose in liver. This point I have to explain. See, what is the sugar produced by plants through photosynthesis?
Glucose.
Glucose, yes, glucose. And in animals, what is the storage sugar? See, whatever carbohydrate you'll take through your diet will be stored in your body in the form of sugar for further energy giving thing, isn't it? So what kind of form? Animal body, for example, human body. Okay, fine. Yeah, here this word glycogen. Glycogen is nothing but the animal starch. The other word for glycogen is animal starch. A synonymous word you can say. So why it is called animal starch? Starch is nothing but the one which will be the storage storage in plants. But when you take animals, glycogen is a form of sugar in which sugars will be stored in the animal body. Okay. So here, when you take the storage sugar in animals, that is called the glycogen. Okay. Now this glycogen will be the storage sugar when we are saying it has to be used when we require energy, isn't it? So when we require energy, this animal starch, it has to be broken down by a process called hydrolysis. Hydrolysis is a chemical reaction where the water molecule, hydro, will be broken out, lysis. So from glycogen, a water molecule will be removed, H2O, and it will get converted to glucose. You got the point? Here, we are having glycogen. Say I'm writing on the top of the slide. We are having glycogen. It should be stored inside the body. When we require energy to do various metabolic processes or physical processes, this stored glycogen will be converted to glucose. Okay. Now, how this conversion is happening? By the removal of water molecule, H2O, water molecule, minus sign I'm showing just to indicate removal. So, this removal of water molecule is known as hydrolysis. So, this would be done because of the smooth endoplasmic reticulum. Okay. Now, where it will happen? It will happen in liver. As we are talking about animals, it will happen in liver. So, a question can be framed, the smooth endoplasmic reticulum is abundant. Understand the word abundant. Abundant means what? You will find in more number in liver cells. For what purpose? For conversion of glycogen to glucose. How? By hydrolysis. What does mean by hydrolysis? Hydrolysis means nothing but removal of water molecule. From where water molecule will be removed? From glycogen, water molecule will be removed. And you will get glucose for energy purpose.